Hello and welcome back to this lecture 22 of Bio Microelectromechanical Systems. Let us quickly review what we did last time. We talked about DNA sequencing using William Sanger's process, wherein we actually have a chain terminated DD NTP set of groups and 4 different reactions. As we know, the DD or the dideoxy NTP is essential for terminating the chain or the process of replication. So you can have various lengths terminated along different specific groups. So when you run all these products of all the 4 reactions along a gel parallel to each other in 4 parallel tracks, you get different lengths which identifies what really is the particular group at that instance or at that length, okay, from the overall readout. So essentially you can actually sequence a DNA by looking at where along its chain the DD-NTP, whether it is an ATP or a CTP or GTP or thymine triphosphate, it is able to cleave and terminate the chain replication reaction. Okay. We also saw a little bit glimpse of the technologies, the most modern evolving technologies where the Sanger's reaction can be used and the transport of DNA could be done on a molecular level using an STM pen or an AFM tip. We further tried to explore DNA nanopores, wherein you basically see how in nature self assembled proteins over lipid bilayers can be used as DNA channels, DNA nanochannels. Similarly, you could also use a silicon on insulator platform to create a thin nanopore, okay, over this platform, and then use it successfully to translate DNA from a rich to a lean side using an electrophoresing potential. Okay. So this can also be an identification protocol for sequencing DNA, as we talked about in details. We discussed some of the other alternate strategies of detection, especially mechanical detection. Whenever you talk about cantilevers, mechanical cantilevers essentially are diving board like structures. So you basically have a change in surface energy causing a deflection delta Z, which is given by Stoney's equation, and then from there you can actually find out whether what is the change in surface stress causing a certain delta Z value, which you can detect otherwise using optical or other means. So therefore this kind of summarizes what we did last time. I would like to now look at proteins. Okay. So we will do some preliminary introduction of proteins today, and then we also like to correlate how this fantastic nano machinery of a living cell kind of translates the information which is available as a compressed DNA or an RNA into an amino acid sequence or a protein. So let us look at what a protein really is. So if you look at this figure right here, a protein really is a bunch of different amino acids. Okay. So what is an amino acid? You have an alpha carbon, you have a carboxylic group here, you have a side chain which can be a polar or a non-polar moiety, and then you have an amine group at one end, and then there is a hydrogen on this alpha carbon. So this essentially is what an amino acid would look like. It is a carboxylic group on one side and an NH2 group on another side of an alpha carbon. That is what the basic idea of an amino acid is. Now the way that these amino acids kind of bond together is through these amide bonds. Okay. So you have this, let us say is one of the amino acids with the C alpha, the alpha carbon right here. Okay. And then this is the amine group, and this is the carboxylic group, essentially the COOH is the carboxylic group on the acid, and the other amide here or the other amino acid here also has another group, let us say R dash on the side chain. Here it is R, and this alpha carbon is connected to an NH2 and also a COOH. So there is a bondage between the OH on the COOH and the NH2 here, thus producing a water molecule, and you have what you call a peptide bond. Okay. So this essentially is something called a peptide bond, C single bond N. Okay. And there is a double bond on one side with O, and on the N side you have a single bond H. So that is what essentially a peptide bond is. So several such amino acids bond to each other with different N groups maybe with these peptide bonds to form arrays of or chains of molecules called proteins. So protein is basically defined as a chain of amino acid interconnected using this peptide linkage. Okay. So this can be essentially a model of how a protein would typically look like when in solution of a certain kind. So one important point about protein is that it undergoes a rapid conformational change because of this R group. The R group, as you see here, can be polar or non-polar depending on the type of applications that, or depending on the type of various amino acid groups that are present in this in this world. Okay. So there are about 20 different such amino acid groups which are categorized into polar, non-polar, ionic, non-ionic, etc. Alright, so essentially let us look at some of the interesting things and the pH dependence of such an amino acid. Okay. So if you really look at this carboxylic moiety here, it is a COOH, right, it is an acid group, carboxylic acid group, and you have an amine group. So a protein when put in a certain pH can absorb an excess hydrogen ion onto its NH forming NH2 forming NH3 plus, or it can depending on if the pH is slightly basic generate a hydrogen ion, thus creating COO minus. What is interesting here is really that it is a function of pH. Okay. So suppose the pH is very acidic, it is less than, let us say, 2, okay, this hydrogen ion concentration. So in this instance, as you see here, because of the excess available hydrogen ions in the solution, there is a tendency of this amine group NH2 to protonate to get into NH3 plus. That is one aspect of how a positive charge can be produced essentially on the amino acid. Now there is also a range of pH. Let us say in this particular case here it is between -2 and 9.5, right. So it is from slightly acidic to kind of reasonably high basicity. In this particular pH range there is a tendency of the carboxylic group, okay, the carboxylic group to protonate. So because there is an excess of OH minus concentration, so therefore there is a tendency of the COOH here to protonate and get converted into CO minus, as you can see here in this case. Now in this kind of a condition also the total amount of positive and negative charges on this particular moiety are equal and opposite to each other. So they are essentially known as zwitterions. Okay. There is a term to denote these kind of moieties where there are ion centers within the same molecule, but then the amount of positive and negative charges are equal and opposite, and so they are called zwitterions. So they have equal number of positive and negative charges. Now if you change the pH into more basic, let us say if the pH goes above 9.5, in that case there is a tendency of this the excess hydrogen ion which was already formulated here in the zwitterionic state to kind of protonate into the solution. So the H plus goes out into the solution because the basicity simply is high enough for any positive ion or any positive charge hydrogen moiety on the amine group to be present. So it kind of protonates. So here again as you see, the overall charge of the amino acid reduces to negative. So the charged state of an amino acid really depends on the pH. So if it is very highly acidic, you have a positive charge on the acid. If it is in a certain range where there are equal amount of positive and negative charges, the amino acid essentially behaves as a zwitterion. And then if the pH is carried on from there to more basic, there is a tendency of the protonation to happen again, and then you have essentially a negative charge on the particular amino acid. Okay. So depending on the side chains or this R group here, okay, this is very critical. The R group may be polar, may be non-polar, and depending on whatever it is or whatever its nature is, there is a tendency of the proteins to change their shapes and sizes by folding. Okay. And essentially this is also known as a conformation change. It's technically known by that name. but the way that protein formulates into a closed loop or a structure is really a function of what are there on the R groups on the protein, or these side chains of the proteins are very, very critical to determining the interaction of the protein in a aqueous or a non-aqueous environment around it. So amino acids can, depending on the side chain, be divided into 4 groups: non-polar or hydrophobic, polar or with some amount of kind of separated charges, positively charged where there is an additional H plus ion on the particular amino acid, or negatively charged where there is an additional minus charge on the carboxylic group by sending out an H plus into the solution. So there are essentially, based on the R groups, we can divide all the amino acids into 4 different groups. So as we already learned that these side chains containing carboxylic groups and amine acid groups is really sensitive to pH based ionization. That is one aspect. Okay, so let us look at what are the R groups really, and if you can categorize all the different amino acids, they can essentially be drawn out as about 20 different amino acids. Okay, so all the proteins that you see around, thousands of chains or thousands of types of chains, are formulated from these 20 basic amino acid groups that are present. Okay, so therefore you can say that you can categorize the side chain of such an amino acid into a non-polar moiety like alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, methionine and proline. So these are essentially the non-polar groups on the amino acids, the R groups on the amino acids. Then you can also categorize the R group into polar amino acids, where this R has a kind of slight shifting of the positive and negative charge centers, although the overall molecule is neutral, but just as it happens in water that there is slightly more electronegativity in one center of the molecule, and depending on which there is a charge separation, here also there is a kind of charge separation within the same molecule. Okay, so that's why polar. So the R groups can be having different names like glycine, you have serine, you have threonine, cysteine, tyrosine, asparagine and glutamine. These are some of the polar amino acid groups. Okay, you can also divide the R groups into negatively charged amino acids, and they can be like aspartic acid or glutamic acid, like you can see in these 2 cases, and then you have also the positive. So here in the negatively charged amino acid, as you see, there is an excess amount of negative charge centers over really the positive charge center. Okay, here also it has a excess amount of negative charges in addition to the only positive charge that you have here. So these are essentially are a function of pH states, various pH states, but there is of course an additional negative charge here. And then you have categories of positively charged amino acids like lysine, arginine and histidine, where you can see there is a negative charge center, but then essentially there are 2 amines which at a certain pH would be able to, even in its deuterionic form, in the true essence or in the same pH range, has 2 charge centers for positive charges and 1 really for the negative charge. Okay, so these are categorized into positively charged amino acids. So in a nutshell, depending on this type of side chains, these 20 different amino acids can be categorized into various kinds like non-polar, polar, negatively charged and positively charged. What is also important here is that let's just do some combinatorials to find out the kind of sequences or the total combinations of sequences that are available to us, which can define the whole protein world essentially. So there are around 20 different amino acids, right? So this is kind of a list of all these 20 different amino acids. There are polar, non-polar, negative or positive charges. You just saw in the last slide about these. These are some of the synonyms which are used either in 3 letters or 1 letter, as you can see here. And essentially the proteins also contain certain derivatized amino acids which are a result of enzymeric actions on amino acids already incorporated. So you have because of some enzymeric actions on these principal amino acids, there are certain derivatized forms, in addition to the 20 basic amino acids. These are kind of represented here, but I am not going to go into this. It is beyond the scope of this course to go into this enzyme derivatized amino acids essentially, but there are a lot of these enzyme derivatized acids in addition to the 20 basic amino acids that we have. So if we want to do a combinatorial, really let us say we are talking about a 25 amino acid sequence, okay, and each of these 25 places can have about 20 different amino acids, okay. So if we can repeat a sequence, then the total number of permutations that we will be having is about 20 to the power 25 moieties. So such a vast amount of arrangements are available only for a 25 amino acid sequence, okay. So you have 20 groups like these, and you can have 25 groups like these, I am sorry, and you can have 20 amino acids in each of these area. So essentially 20 into 20 into 20 so on up to the 25th term. So 20 to the power of 25 combinations. So this is a vast amount. So if you wanted to predict what is the one sequence out of these 20 to the power 25 sequences of amino acids, it is really a huge number to deal with, okay. So these are essentially the primary structures of the proteins, and I am just going to come to that in a little bit. What I would like to show here is that the way that proteins would actually be formulated are in a sense a polymerization process, wherein two amino acid groups are condensed together in an enzyme catalyzed condensation reaction. Why we call it condensation is that wherever there is a disbursement of a H2O moiety or a water molecule, we call that reaction a condensation reaction, okay. So here this process is enzyme catalyzed. So there is an enzyme which would be able to formulate the peptide bonds between the two amino acids, okay. Now this is a very important concept which is very well applied to even nature, where mother nature tries to replicate the amino acids from mRNA sequence in the translation process, okay, where they also use enzymes to cleave bonds or to make the peptide bonds. So it is enzyme catalyzed condensation. We already know that the H2O moiety goes out, one on taking the OH from the carboxylic on one and H from the NH2 on another, right. But this process happens only in the presence of an enzyme, okay. So a peptide bond is formulated. So here in this illustration it pretty much shows how this is done. So you have two amino acid sequences here, and then there is an enzyme which would convert this, and there is a condensation reaction. So there is a H2O molecule which is eliminated, and you can have polypeptide chains like these, wherein you have several of these peptide linkages and more than one amino acids. So proteins can be this large. So molecule therefore, depending on how many peptides the molecule would have, is classified either into a dipeptide or a tripeptide, tetrapeptides, so on and so forth, and that also determines how many amino acids you have. So one protein or one peptide linkage is associated with two amino acids, okay. So you could find out the total length of the protein chain based on and categorize into a di or tri or tetrapeptide or so on and so forth, okay. So we already know that how many combinatorials can be processed possible only in a 25 amino acid sequence of protein. This small sequence of protein can have about 20 to the power 25 combinations of if you can club all these possibilities of each place being filled by 20 different amino acids, okay. So changes in sequence of a protein can lead to significant changes in the protein structure and the functionality. So if the sequence is altered slightly, it's varied slightly, you can have altogether different structure of the protein of interest. What is also important here is that if we really categorize proteins into different kind of structures, they can be classified principally into the primary, secondary, tertiary and quaternary structures. And let us just investigate a little bit about why they are called so or what are the differences between. So this cartoon here illustrates very nicely these 4 different structures. So proteins do not really exist simply as a long extended chain of amino acids. It's hardly possible to exist in a natural state in a long chain. One of the reasons why that is so is that because there are a lot of OH groups, and also there are a lot of side chain interactions, and there is also a tendency of hydrogen on the NH2 or COH, the OH on the COH, to interact with the solvent where it is in, and so there are additional hydrogen bonds which are created in between the molecule and between the molecule and the solvent in question. So conformation really is a very dynamic process. The overall structure that happens is a very complicated structure just because of the several hydrogen based interactions between the protein moieties and its surroundings, or even molecules within the same protein moiety. Okay. Now primary structure though is very important in understanding what is the sequence of the amino acids on the proteins. So essentially primary structure is the basic foundational structure. It gives a kind of relationship between what follows what in terms of amino acid sequences, although it does not exist in nature, but it is always wonderful to represent a protein with its primary structure to find out the amino acid sequences that it has. So interactions between of course the amino acid chains would cause the proteins to bend and fold. Okay. So interactions happening between the amino acids cause bending, folding, coiling of these chains, and eventually what emerges is a specific 3-dimensional structure, and this is known as conformation, as I have been discussing before. So secondary structure, as you can see here in this illustration, is really formulated within localized domains. Okay. So because of course because of the presence of hydrogen bonding, and particularly because there is a carbonyl group and a peptide bond. Okay. So in between the protein backbone itself there is a lot of bonding because of these 2 moieties. So peptide bond on one side, and then maybe the carbonyl group on the other side. So secondary structure is like kind of coiling like this. So you can say that if you leave this is a very fine analogy that I keep giving, that you have a straight let us say hose pipe. Okay. And this was otherwise coiled, this hose pipe was otherwise in a coiled manner, and coiling is by means of either stressing in a heated environment or something. Now whenever you stretch it totally in tension and put it in tension and stretch it, this is the primary structure that it would have. But the moment you release the 2 ends, it is going to recoil back. Okay. And because the stresses which were there would try to align it back into the equilibrium, and that is the secondary structure. And when you drop this, let us say this is a telephone cord we are talking about. Okay. So now you have a case where this you have just about released enough so that the structure gets coiled, and then you drop the whole structure. So what would happen is that this coiling would start to bend among itself, and there would be an interaction between the coiling itself. So that is what the tertiary structure of a protein would be looking like. So there are interactions which are beyond the local domain. Okay, but they are essentially confined to the same molecule. There is only one single molecule. There are interactions between different domains of the molecule rather than just adjacent interactions as were happening in the secondary structure here. So this is what the tertiary structure of a protein is. And then of course, so formulated because of interactions between side groups of the chain in different regions of the same molecule. Okay. And then there is this quaternary structure which is essentially referring to interactions between subunits or individual polypeptide chains between multi-chain units. So here for instance there is one protein which is present here in this area, and another protein maybe which is present independently in this area. So there is always going to be, so there are 2 tertiary structures present close by. So there are going to be interactions between the 2 structures. Okay. So therefore between subunits probably of the same protein or individual polypeptide chains all laid side by side in a solution, those kind of multi-chain interactions develop what you call the quaternary structure. So therefore, starting from a simple to a very complex, proteins have increased degrees of complexities in their conformations or in their shapes and sizes. So if you look at the proteins in general, although there are many structures of these proteins which are possible, we can principally classify the base units of these structures, just as we have let us say different unit cells in a certain lattice. We can also classify the proteins in terms of some base units, a repetition of which would essentially build the molecule as good as good in the crystal structure. As you know, the repetition of the unit cell in the 3 dimensions makes the whole molecule. Okay. So there are 2 principal structures which are foundational for formulating or demonstrating the overall complexity in the protein structure, and these so called analogues of unit cells are known as the alpha helix and the beta pleats of proteins. So you can either classify into an alpha helix structure, and I am just going to come in a little bit into what an alpha helix really means, or there is a beta pleat where you can say that the protein is kind of rotating and the molecule is like a big multiple S's together compressed. So there are parallel sheets of amino acid chains which are running at different levels. Okay. So in alpha helix, as you see right here, you can see an alpha helix here in this region. Okay. This is an alpha helix really. So so each peptide here forms hydrogen bonds with the 4th amino acid above it and the 4th amino acid below it. Okay. So the way it goes is that this peptide linkage, which is essentially this C N double bond O and H here, and then you have the other portion of the protein in both sides here, the alpha carbon and things attached to it. So here you have NH2 attached, here you have a COH attached, something like that. So this peptide bond with the 4th amino acid in the sequence ahead and the 4th amino acid in the sequence back there formulates hydrogen bonds, because of which there is a tendency of this molecule to kind of form alpha sort of structures. Okay. And this is on a 3-dimensional basis that it is formulating the alpha. So it is more like this. Okay. It is more like this. That is the alpha structure that we are talking about. So essentially so it kind of coils just as any telephone cord would do. Okay. And there are bondages formed between the peptide and every 4th amino acid on either end. So 4th amino acid on the bottom, 4th amino acid on the top, they would have hydrogen bonds. Essentially in one helical unit or in one unit of the helix that is formulated, there are about close to 3.6 amino acids in this kind of an orientation. Hydrogen bonds would define the alpha helix in a manner where in one helix you have only about containment of 3.6 amino acids. Okay. So I mean there is no physical interpretation of the 0.6, only that 60% of one full amino acid is within the chain in additional to the 3 units. That is how you define the helix length, okay, of this particular structure. So also one interesting factor here is that the hydrogen bonds are kind of parallel to the axis of the helix. So that's the reason why this alpha kind of structure is formulated. This elliptical or this I would say this helical kind of structure is formulated because all the bonds essentially in all the different areas of the proteins are parallel to the central axis of the helix, which confines the molecule within this helical domain. Okay. So that is what alpha structure is. So the second kind of like the analog for unit cell you may call, or the second kind of the structure that is most commonly used as a unit structure in protein are the beta strands. The way these beta strand structures can go is that they are like parallel sheets or they are like anti-parallel sheets. There are two different kind of beta structures where these proteins would be laid on the top of one another like sheets of different amino acid sequences, as you can see here. These are parallel because as you see in both the molecules here the groups are kind of together. This is the C double bond O, right, this is the NH, okay, this again is a C double bond O, this is again the NH. So you have parallelity in the way these groups are placed on a single sheet of this beta sheeted structure. So that is why parallel beta sheets. You may have an anti-parallel configuration where the C double bond on one would comprise or would face NH on another or vice versa, okay. So essentially this is an anti-parallel kind of arrangement. So therefore polypeptides can extend as zigzag chains, as you are seeing here, okay, called beta strands, and the region of the polypeptide with an extended chain, the conformation is known as a beta strand. Beta sheets are stabilized by hydrogen bonds, as you are seeing here. There is a hydrogen bond here between the C double bond O on the carboxyl and the NH on the amine group. Similarly, there is another one here between the C double bond O on the lower sheet and the NH on the upper sheet, and so on and so forth. So there are a lot of hydrogen bonds, although whether in parallel or in anti-parallel. Of course the anti-parallel has a lot more stability than the parallel has, okay, and so therefore they can be parallel or anti-parallel depending on whether each section of the chain runs in the same or the opposite direction. And the beta sheets are stabilized by hydrogen bonds between two or more beta strands, okay. This is the anti-parallel beta sheet, this is the parallel beta sheet. So in a nutshell these are the two structures, alpha helix and the beta sheets. Now after going to this little bit and understanding the mechanical aspects related to the molecular structure, I would like to go into a very interesting area which is also known as DNA transcription and translation processes. Why it is needed is probably one of the most fascinating ball games today of chemists as well as molecular biologists, because this in a sense gives the cell its stability, its physiological stability. So there is really, it is like a genetic code, it is like a small computer, the cell, which can convert a compressed code of these ATCG sequences into a sequence of amino acids or proteins. But the proteins, as we know, are more or less present in the cell membrane, and which is also responsible for the state of health or the physiological state of the cell. The way it grows, replicates, everything is related to the surface proteins that come on the surface of the cell. And the process which allows these proteins to be generated is situated deep within the nucleus of a cell, where the messenger RNA is essentially formulated from a section on the DNA, a section on that small region in the chromatin and the nucleus, but there is a compressed sequence of helical coils of DNA which we also know as chromosomes. So there is a process within the cell where the DNA molecule gets converted into a messenger RNA molecule, and this is the compression algorithm, okay. So the amount of data you have on the DNA is certainly much bigger in comparison to the amount of data you have on the mRNA. It is a much shorter information sequence than on the DNA. So you have a compression algorithm here, so you have a messenger RNA, and this process is known as transcription. So you are making a transcript and you are not changing the language really. The language is the same as ATCG, only in this case the T, the thymine, is replaced by an uracil, as is normally the case with all RNA moieties, ribonucleotide moieties, okay. And of course this is a single stranded structure which is formulated from the DNA, which is a double stranded structure. Now the mRNA gets generated within the nucleus, and then it goes all the way up to outside the nucleus into a place called the endoplasmic reticulum. It is an organelle which is there inside the cell, and it is essentially called the protein warehouse. So there are these intelligent nano machines called ribosomes, okay, on the surface of this organelle of the cell, which essentially would take up a mRNA sequence and then convert that into a protein sequence, okay. And the way it does is very fascinating. There are some amino acyl transfer RNAs which are moving around in the solution, and there is a group to group matching or a mismatching which leads to the amino acid sequence as the mRNA or the messenger RNA sequence is read at the nucleus level, and thus the amino acid sequence of the protein is formulated by this process of translation. Why it is called translation is because the language of the mRNA, which was more in terms of A, T, C and U, or sorry, A, C, G and U, is now converting into altogether different language. So you are translating the information on mRNA into a sequence of amino acids. So it is a language conversion that is happening. So that would result in development of these surface proteins here, and this is a function of how the cell would stay in a certain environment. So the way that a cell reacts by having a signal from its environment would be in turn expressed by or would be in turn kind of try to be back into so the cell will try to be the cell will try to be in equilibrium based on whatever responses or stimuli it is getting from the environment, and the way that the equilibrium happens is through producing or production of a set of proteins which would exactly cause the deviation from the equilibrium to be balanced back, so that the cell comes back to the equilibrium. This depends on what is the existing state, physiological state, and what is the deviation from that state, and what would be needed to control that deviation from that state. These are the three steps which defines how the transcription and translation process would produce these mechanisms which would cause this equilibration to happen again, known as proteins. Okay. So let us look at this in a little more details. So in a nutshell, transcription, where double stranded DNA, you can see this DNA here getting converted into a single stranded DNA, right, and the enzyme that so you use an enzyme for this conversion, and this is known as the RNA polymerase. So it synthesizes the messenger RNA, okay, as just in here. So the RNA synthesis done by RNA polymerase molecule from DNA to RNA, where a much more compressed code is developed based on reading the DNA molecule. And then you have these translation which is done by these intelligent nanomachinery called ribosomes, which translates the sequence of the bases in the mRNA to the corresponding proteins, and these proteins then perform various functions inside and outside the particular cell. Alright, so this is essentially the amino acid sequence which you develop from this mRNA here, which has been developed further from the DNA. So let us look on a step by step basis of how this whole process can be accomplished. Okay, so this is essentially the transcription process as you are seeing here. Let us first talk about what exactly it is all about. So this is a double stranded DNA structure that you can look into, right. This whole structure here is a double stranded DNA structure. This is the corresponding RNA structure that has been formulated. The only difference between a DNA and an RNA is that an RNA is a single strand, okay, as opposed to DNA, and then the group thymine here on the DNA is kind of replaced by another group called uracil on the RNA. So the 4 bases in ribose sugar RNA are ACUG as opposed to ACTG in the DNA. So the process is essentially starting when the cell copies a particular portion of its DNA nucleotide sequence into an RNA nucleotide sequence. I am going to look into the details of how this happens in the next slide. Okay, the information on the RNA, although copied into another chemical form, is still in the same language, and therefore it is known as writing the transcript or transcription of the DNA. The name transcription is because the language is same essentially. There is no difference in the language of the DNA or the RNA, except that there are a few groups here and there which are different, and also the very fact that this RNA is a single stranded structure or a single stranded molecule. Okay, so we use an enzyme here which would be able to read what is there on the DNA and produce compressed information as an RNA. So this enzyme is known as RNA polymerase, okay, and this is used as a catalyst to catalyze the transcription process. So essentially the RNA polymerase is nothing but a set of proteins, okay, and it contains about some protein moieties and some RNA molecules in turn. So here itself this kind of gives the complicated structure of an RNA polymerase. Okay, and you can see here in this particular region that the DNA chain is kind of opening and coming out, and then there is an alternate RNA chain which is evolving from here. So there are several steps which are used by the RNA polymerase to carve a messenger RNA from a DNA, and I am actually going to just look into the mechanical aspects of this process step by step. So what you need is that you need to bite into the DNA at a portion where the signal which, so there is a signal which comes into the cell. Okay, a signal may be in terms of a chemical group or a moiety which can be able to identify a portion of the DNA from which you can replicate the corresponding mRNA, so that eventually after the translation process on the endoplasmic reticulum, the mRNA sequences out a protein which is useful for equilibrating the state of the cell back to its normal. Okay, so there is a deviation in the environment of the cell, due to which there is a change in equilibrium of this particular cell, okay, due to some reasons, and then there is a signal which is generated because of this change in equilibrium, which goes and bites on the portion of the DNA from which you are essentially trying to build up the messenger RNA. Okay, and then once the messenger RNA is built up, it produces a protein which would go into that changed environment and try to bring it back to the equilibrium position or equilibrate the whole cell. That is the whole fundamental of this transcription translation process. Okay, let's look at how. So the RNA polymerase catalyzes the linkage of ribonucleic nucleotides. Unlike DNA, RNA does not store permanently the genetic information in cells. Okay, so some characteristics that the RNA polymerase has first have to be evaluated for understanding how the RNA polymerase will do this translation, this transcription work. So unlike DNA, RNA polymerase makes about 1 mistakes in every 10 to the power 4 nucleotides while doing this. DNA it is about 1 in 10 to the power 9. Okay, so this one mistake in every 10 to the power 4 nucleotides would mean that there are several mistakes that the RNA can do while getting replicated from the DNA, and so it is not that vital or that important as the replication of a DNA process. Essentially, replication of a DNA would cause a mutation. It's a permanent change resulting from this information change on the DNA, whereas there are thousands of RNAs which are produced at any point of time from the DNA structure, and therefore it is not that critical to have a very low, very high fidelity or very low error rate essentially. So you can manage within about 1 error in every 10 to the power 4 nucleotides easily, and still nothing appreciable changes. Essentially the protein coded from this mRNA would have a slightly different sequence, and then that really is not very critical because there are several of these proteins which are being produced every second by the cell. Also, the consequences of an error in RNA transcription are much less significant than in DNA polymerization, as I just talked about. Okay, so RNA polymerase has a modest proofreading mechanism. If an incorrect ribonucleotide is added to a growing RNA chain, so you have an RNA chain which is growing on the basis of the template molecule of the DNA, then the polymerase can back up and the active site of the enzyme can perform an excision. So therefore there is a cleavage of the inappropriately added nucleotide as and when the RNA polymerase is able to sense it. There is a confirmatory change, and based on that there are many types of RNAs which could be created, ribosomal, small nuclear transfer or messenger, different forms of RNA. So this here illustrates how this process happens. So this structure here is essentially the RNA polymerase enzyme. Okay, I actually call it a magician's hat. So here there is this chemical signal called and this is essentially described for the bacterial cells. Okay, so this transcription process here that I am going to describe in just about a few minutes of a few seconds is in bacterial cells. So there is a chemical signal which would come to this particular RNA polymerase enzyme, which would tell about where exactly on the DNA you should be able to bite, so that you can start producing the messenger RNA. Okay. So this chemical signal is also a function of the environment. So it is called sigma factor. Technically this is the name that is given to this chemical signal. So this chemical signal comes and it kind of sits on this RNA polymerase, and the RNA polymerase essentially goes into reading the DNA. So this is a DNA molecule here, as you are seeing, and the RNA polymerase kind of goes into reading the DNA through its sigma factor. So we have to find out first the site from which the transcription process should start. The RNA polymerase should be able to start the transcription, or it should be able to know where to start the coding into mRNA from the DNA, okay, the transcription. So let us suppose with the sigma factor the RNA polymerase kind of bites into this DNA, okay, this bites into this DNA region, and then it loosely slides on this DNA from this end all the way up to an extent when it hits a so called promoter region. This promoter region on the DNA is a kind of chemical orientation and structure wherein the RNA polymerase and the sigma factor especially would be able to bite on the DNA molecule. Okay, it is able to bite on the DNA and open the chain. So that is called the promoter region of the DNA. So let us suppose this is this DNA chain, and this enzyme along with this sigma factor is kind of sliding past. So the enzyme is an upper and a lower half, and you have the DNA chain which is going into this and coming out of the other area, and it kind of slides closely over this until it hits up a promoter region where it kind of bites and holds back firmly, so that it opens up the chain of the DNA. It is a double stranded DNA, right. So it opens up the chain of the DNA. Okay. So therefore, if you really look at this process, you have a detachable subunit, sigma factor. It directs the polymerase to a site where it should start the transcription, and the RNA polymerase molecule only weakly adheres to the bacterial DNA when they collide with it, and then the polymerase molecule slides over rapidly along the length of the DNA molecule until it hits the promoter region, okay, and there it kind of binds firmly or more firmly over this specific region because of the sigma factor. So when the polymerase slides over the specific region called promoter, it binds firmly with this, and by binding we are meaning we are trying to cut the DNA chain open into two strands. It is removing the hydrogen bonds between the base pairs on the DNA and cutting it into two different strands. So this starts the initiation of the RNA transcription process or the RNA coding process. So once this has happened, as you are able to see here, when the sigma factor has gone and found the promoter region and it has been able to open this chain, so the DNA is open chain here. So when it comes out on the other side of the RNA, it again kind of chains back or zips up, okay, because thermodynamically that is the most favorable state once it goes outside the biting mechanism of the RNA polymerase. However, in the region where the sigma factor has kind of bitten closely on the DNA, there is a chain opening like this, and these two are open, from which one half of this chain would be used for templating the other half of the molecule. So you are using this lower half, let us say, for producing this as a template to sequence the mRNA or the messenger RNA in the reverse sequence of what is there on a DNA. So therefore, we can think of a compression of information. This model is something where we can illustrate this, or we can define this as a we do not need all the information that is there on the DNA, but we really need a selective specific region of the DNA which would be able to code a protein, and that signal, when it comes, it kind of automatically compresses all these informations into a really useful domain. Okay. So it is a kind of signal compression on how many base pair starting from what point and ending to what point is really needs to be transcripted as a messenger RNA. Alright, so the binding action opens up the DNA helix here and exposes the short stretch of nucleotides on each strand, and so therefore there is a templating mechanism which has started taking place now. Okay, so whenever the two open strands act as complementary templates for the incoming ribonucleotides, two of which are joined together into an RNA chain by the polymerase enzyme. So the polymerase essentially furnishes by assembling the ribonucleotides which are continuously coming in that, so there are a lot of ribonucleotides which are going around, okay, in the solution in the nucleus. So they continuously keep on diffusing through the RNA. So as and when suppose there is a C group on the opened up DNA, it should correspond to a G group on the nucleotide. So there is essentially a capture point for a G to have come and suitured there. Then there is a, let's say, a following group on the parental RNA which you are, or in the parental DNA which you are trying to copy, where there was C before and now there is a G following C. So correspondingly there would be a C nucleotide which sits next to the G nucleotide, and these are individual nucleotides. So the polymerase, what it does is it suitures these two nucleotides, and so therefore it kind of starts zipping the DNA in the internal region where it has already opened up. Okay, but this zipping is action is by the individual nucleotides which are now trying to kind of get templated or arranged by this template DNA molecule. Let's say, I'll just illustrate this a little bit more clearly here. Let's say you have a DNA which has opened up, the chain has opened up like this, right, and you have a template sequence here which is corresponding to A, G, C, T, and then you have again a C, G, something like that. So there are a lot of nucleotides which are rushing in here. So let's say this site has been identified as an A, definitely a nucleotide which would be independently, let's say, uracil. Okay, we know that in an RNA there is an uracil, so therefore there is no thymine in this region as an independent nucleotide, it is more in the uracil form. That would sit on the A. Then there would be a C or a cytosine which sits on the guanine here, and then the RNA polymerase essentially has a role in bonding these two. Therefore there is a guanine which sits on the cytosine here, a hydrogen bond, and the RNA again suitures this C to G. So this RNA molecule is slowly developed on the template here that you can see, and this is what is happening inside the RNA polymerase enzyme. Okay. So really after the first 10 nucleotide sequences are kind of synthesized, the sigma factor relaxes itself by holding it less tightly now. So it kind of ungrips or loosens out its grip on the DNA. So there is no more a question of cutting open the DNA. You have already initiated a zipping up process, and then it is thermodynamically favorable to kind of keep zipping up as the RNA polymerase kind of rapidly moves through the DNA chain. So really after the first 10 nucleotide sequences, as you can see here, the sigma factor no longer plays a role of biting. It is the RNA polymerase sliding and the auto opening of the chain which promotes this process, and continuously the nucleotide sequence as the mRNA is being generated from this template here that I showed just about a few minutes back, which has been opened up inside this whole RNA polymerase structure. Okay. Now so therefore the chain elongation continues, and there is of course a rate aspect to this elongation. Normally typically this suturing rate and the nucleotides getting assembled over the template would take close to about 50 nucleotides per second or so. So this is a finitely definable rate of production of the mRNA. So the speed of production is about 50 nucleotides per second. So now once the process has started, of course mRNA is a size defined molecule. There has to be some kind of a signal to the enzyme which lets it know that it has to stop and it has to dissociate the mRNA out, and thus closing the template. As you are seeing here, the DNA template which had zipped up or which had been zipped open in this region, as you are feeding the template within the molecule was actually zipped back into or zipped close as it goes across the RNA and tries to emanate out of the RNA, right. So, this is the point from which it is emanating. So, the zipping up again occurs when it emanates. The only open region is inside the RNA polymerase. Let us say this is the RNA polymerase molecule. The only open region is inside this RNA polymerase molecule. Now, you have to somehow let the RNA polymerase realize that the sequence that it has to make is complete and it should no longer produce any mRNA. Okay. So, there are sequences on the mRNA which are also known as stop codons, which kind of there are few sequences, at least three different kind of sequences where a combination of nucleotide is kind of hit upon, where there is a conformation change of the enzyme, the RNA polymerase, as it hits upon this 3 base pair, uniquely combined 3 base pair region called the stop codon region of the mRNA. So, whenever that happens, so this is essentially also known as a terminator sequence, where the chain kind of terminates from getting manufactured. So, essentially this is a combination of 3 nucleotides on the mRNA, on the produced mRNA on the template, where the RNA polymerase kind of gets a conformational change or a jerk, because of which there is a seizure of this templating activity. The RNA that is developed, the messenger RNA that is developed on the template kind of detaches from the template. Okay. So, typically there could be mechanisms like, let us say the RNA molecule hits upon some kind of a hairpin loop like structure here. So, the molecule that has just emerged because of this stop codon, in that region it formulates some kind of a hairpin loop. And so, because this loop has been formulated which is not normal or not a normal sequence, there can be a sudden jerk or a conformation change which can also cause this detaching movement to happen. Whatever it is, the change in conformation of the RNA polymerase is what we are wishing to have, so that we can terminate the the chain fabrication process. So, that is exactly what happens here. It forms a hairpin loop and it tries to wedge open the movable flap on the RNA, and so it releases the RNA from any further synthesis. Okay. And so, the free polymerase is welcome to pick up another sigma factor and start the DNA sequencing accordingly on a different section of the DNA. So, the whole idea is that the proteins which are being produced are a function of what sigma factor or what chemical signal comes into to bite a certain region, from which all it needs to hit upon is a terminator sequence for defining the length of the mRNA that we are producing. So, it is a very vital and interesting process of information compression that even the cells or the nucleus on the cell with this intelligent molecule RNA polymerase can execute. So, this kind of brings us to the end of this lecture. I would like to illustrate this concept in a little more details, and also start with another very important translation topic, which would give us the conversion between the language of the DNA or the language of the RNA into the language of amino acids or proteins. Thank you.